Hi, everybody. Welcome to GymB Live. I am Rachel Horak. I'm in the ASM Education Department. Today's paper, we're going to talk about specifications grading in a cell biology course. First, before we get started, I would encourage all of you to take a very quick demographics poll. This is completely anonymous if you chose to, and just give us a click, click, click. Tell us where you're living, if you ever published teaching scholarship, and where did you hear about this event? Right. Thank you so much for giving us some feedback. We really do appreciate it, and welcome everybody. So the format of today's event, participants will be able to ask their questions using the Q&A feature in your Zoom toolbar on the bottom part of your Zoom screen. You will not be able to turn on your mics or your cameras for this webinar. If you have any technical issues, you can put a message using the chat function and send a direct message to myself or ASM:JustinBambler.com. We'll be able to assist you. We are recording this webinar, and the webinar will be posted to GymB's YouTube playlist if you want to watch it at another time. So now I'm going to introduce Stanley Malloy. He's the GymB Editor-in-Chief. Stanley.
Hi, everyone. I'm going to be the moderator for the question and answer sessions today. This is a really interesting paper that deals with this issue of how you test and how that can influence the success of students in a class, and what that might mean for the way the rest of you who are listening in might think about how you would teach your classes in the future. So we're really excited to get started. I'll let each of the speakers introduce themselves, and I know the title of the paper is in the first slide. So Shoshana, I think, are you going to kick us off?
I am not going to start the slides, but I will get them going for Dr. Janelle Talley, who will. Just give me 1 second.
All right. And Stanley, did you want us each to introduce ourselves before we start talking? I just wanted to make sure.
It would be really nice if you each introduce yourself. your name and what you do. Okay.
Yeah. So I'm Janelle Talley. I work at GGC, and I'm an associate professor there. I teach intro bio, genetics, cell bio, biochemistry. Those are kind of my things.
I guess I'll pop on and go next because I wasn't paying attention. A good student, right? I'm Shoshana Katzman. I'm also at GGC, been there about 6 years teaching cell biology, immunology types of courses, and looking forward to present this stuff to everybody.
Jennifer, you want to introduce yourself?
Yes. Hello, I am Jennifer Hurst Kennedy. I'm also an associate professor of biology at GGC, and I've been here about 10 years, and I mostly teach cell biology and biotechnology courses.
Mary Beth?
Hi, everybody. My name is Mary Beth Anzavino. I am an associate professor of chemistry, not biology. I got involved in the project because my background is in chemistry education research. And so I worked on that part of the sort of evaluation and assessment part of the project.
Okay, so I guess I'll go ahead and get started, if that's okay. All right. So I'm really excited to be here, and thank you so much for inviting us to do this talk. We're going to talk to y'all about this type of grading that we've instituted in our cell biology course called specifications grading. And we want to talk to you about kind of how we did it, and also the effect that it's had on student learning and student attitudes in our courses. So to go ahead and get started on that, I'm going to talk to you a little bit about some instructional approaches that we all probably use, or I hope we all kind of use, to foster student learning and retention. And this is not an all-inclusive list. It's just some things that have been shown to work. So these are active learning strategies. This is really, instead of standing up and lecturing and having students just sit and, absorb everything, it's getting them moving in the classroom, using their hands, talking to their peers, using the information that we want them to know, and actually using it in context. Also giving timely feedback to students. So once they're doing an assessment in class, or if it either at formative or summative, that we give timely feedback, that we tell them what they're doing well and why they're doing a good job, or what they need to improve on. So what are strengths and weaknesses to what they've given us? And then another way to kind of help them learn and retain information is reducing stressful, high stakes assessments. So, many of us may historically have given a couple big chapter unit tests each semester for our students, and that can be very overwhelming to students. So are there ways that we can reduce this type of stressful environment for them so that they can show us what they really know, instead of it being kind of murky behind all the stress that they might have coming into that assessment? And then another instructional approach that you can use is something called mastery learning. And mastery learning, really what the concept behind this is, Bloom kind of introduced this in the late 60s. And really this is to help ensure that students are meeting whatever that bar is before they move on. So I know historically for me, when I used to take a test, I'd be like, okay, I'm done with that material. Even if I didn't master it or understand it, whoosh, everybody moves on. So we're just going to keep going ahead no matter what I understand about that previous material. Mastery learning says that's not good enough. Let the student go back and make sure they really understand it. So give them that time to ensure they actually are reaching that bar. Okay, so the next slide. So the framework for mastery learning, I kind of think about it in two parts, kind of what the faculty must do and then what students kind of have to have. So what faculty must do is they need to break down learning objectives or outcomes into more manageable, smaller, specific units. And even within a particular learning outcome, as the teacher, I might have this statement, this is what I want you to be able to do. But the student may not understand all the little pieces that I think go into that particular learning outcome. So I need to state what that learning outcome is and then tell them specifically, within this learning outcome, I want to see this A, B, C, and D. And they need to kind of know what that breakdown is so they understand what's being expected of them. They also, faculty must provide very targeted and timely feedback. So not just taking off points, but why, what about that particular answer, what they provided to you isn't correct or what is correct. And again, making sure that they get that feedback within a shorter time period so that it's still fresh on their mind. Faculty also must provide additional learning opportunities to help students address any deficiencies that they do have. So having like a stockpile of extra problems or different ways that they can engage in that material to address those specific deficiencies that they have is really important. Students, on the other hand, once you've broken down those learning outcomes, they have to have an understanding of what that expectation really means. So they have to be willing to come to you, and if they don't understand, say, hey, what do you mean? I don't really understand what you mean by X, Y, or Z. So they have to be willing to kind of talk to you and engage with you about those learning outcomes, but we have to be very clear about what those are. They also need to have time to revisit their deficiencies. And this, I think, is really important in mastery grading. For me, this is something that was a challenge, but also once we started figuring out how to do it is really, really important for the student. This is the part that allows them to say, oh, you're gonna let me have more time to figure this out. I am gonna take ownership of actually trying to figure it out for myself. And then finally, they have to be encouraged to work with their peers or others, it can be you too, to identify what they're good at and what they need to work on. And this is really important too. So having lots of different types of assessments or activities that they can do that help them figure out what they're doing well and what they need to improve on is really central to mastery learning. All right, so then the next slide. And so one of the ways that we are engaging mastery learning, one of the ways you can do this is using something called specifications grading. And specifications grading, that's what encourages mastery learning. And so what this is taking all those learning outcomes that you developed and having smaller assessments that address each one of those individually. So I might have an assessment, whereas before I would have, an hour and a half long, they need to take an hour and a half or an hour long test. Now their assessment might be 15 to 20 minutes over a particular learning outcome that I've told them I'm breaking down into these 3 or 4 parts. So I have a question that's specifically associated with each of those principles that I want them to know from that particular outcome. So it's much lower stakes. And then the feedback that I provide them is specific to each of those little pieces. So for me, for example, for a learning outcome, I have like a little sheet of paper that goes with each learning outcome, and I've isolated each concept within that outcome, and I'll circle it and tell them this is what you missed, and I give them that sheet of paper back after their quiz, so they know exactly what they were missing and why. And then finally, again, really important, students have the ability to earn extra chances to do the assessment again. So let's say they didn't reach the bar they were supposed to reach. So we give them that feedback, and then they can come and say, hey, I want a chance to try again. And we go, great, here's an additional opportunity. Here's something else that will help you work on this material. You do this, give it back to me, and you earn a chance to try again. And that's really, really, really important for specifications grading, so that students get that extra chance. So now Jennifer will take over for the next step.
All right. Hello, everyone. So to give you a little bit of background about why we chose to do specifications grading in this particular class, a lot of it has to do with the unique position of cell biology within our particular curriculum. So in the figure there, you have one of our curriculum maps, and you see, of course, not all the courses, but the main progression of chemistry and biology courses that our majors would have to go through. And what I want to point out is you see we have cell biology as kind of the right on the cusp of finishing up sophomore, heading into junior year. So while it's not a traditional gateway class like your intro biology series, intro chemistry series, in a way it does sort of serve as a gateway because it is the prerequisite for all of our 3,000 and 4,000 level classes. So we feel as faculty teaching this course that we have a strong obligation to make sure that our students are learning what they need to know in our course so that they are successful throughout the rest of their curriculum. So we're hoping, and we did show that with specifications grading, we are increasing their knowledge, also building that strong foundation, study skills that they can then take with them as they move up into their upper level classes. Great. So as you heard from Janelle, one of the major parts of mastery learning, specifications grading, is taking this material and dividing it up into very clear, very discrete learning objectives. So when we first started working on this project, the team of cell biology faculty, we all got together and we looked at all of our learning objectives for the entire course and worked on making sure that all of our language was very specific and that it was clear and very, very narrow learning outcomes. And then we associated each one of those learning outcomes with a specific quiz. So we broke our content into 20 learning objectives. So this meant that the students each had a quiz associated with that learning objective. So 20 small quizzes, as opposed to the traditional 3 or 4 standard course exams with a final exam. So this, we had the 20 quizzes. And we graded them for mastery on a pass-fail basis. So if a student achieved a score of 80% or better, that's considered a pass. They demonstrated mastery, they're able to move on. Whereas if they scored below a 79%, that does not demonstrate mastery of the material. So they would have an opportunity to retake it. And again, as Janelle said, we want to make sure that the students aren't just like running in to take the quiz again without preparing themselves. So it is really important that they take that time to reflect on what the deficiencies are in their learning so they can prepare and be more successful on their retake. So we would give them some sort of assignment to help them practice the material. So providing worksheets with extra practice problems, the opportunity to meet with faculty to discuss areas they're confused about, or chapter summaries. So we've all kind of done a variety of that. So the student has a total of 3 attempts for each quiz. The first one's given in class. Again, if they score that 80% and mastery, we can move on. But if they do not show mastery, they have 2 additional attempts to take for that quiz after they've done the additional assignments to strengthen their understanding of the content. Thank you. So with our 20 quizzes instead of the traditional exam, this is what the grading scheme ends up looking like for our course. So with a total of 20 quizzes, if students pass 18 or above, that's 90% of the content mastered, that's an A. 16 or 17, that's 80% of the content mastered, so that would be a B, and so on and so forth. So for our particular courses, students need to earn a C or better in order to use this course as a prereq for their upper level courses. So the minimum number of learning objective quizzes they would need to pass and demonstrate mastery would be 14. Okay. And now I'm going to pass things off to Dr. Mary Beth Anzavino, who's going to show us all the data and the statistical analysis.
All right, thank you. Okay, so as instructors, as people who teach content to students and help students learn content, we want to know what effect is this intervention having, right? What effect is this change having? And so we had a couple of hypotheses. The first was that more students would successfully complete the course. And the reasons for that hypothesis are things like the students have multiple chances to show that they understand the material. We've removed some of the stress from the assessments. They're much lower stakes. If a student's having a bad day, it's okay. They always have the opportunity to retake the quiz again. And then the third component is, again, that timely feedback. If students are getting really timely and targeted feedback on the specific things they need to work on, that can help them go back and remediate where there might be some gaps in their understanding, and then come in and take the quiz again and hopefully pass it after completing some extra studying in that area. Our other hypothesis related to student attitudes, and we thought that the students would, in the specifications grading courses, would have more positive attitudes towards their grades and level of understanding of the course content. So I'm going to talk about the first hypothesis. We're going to take a look at some grade distribution data, and then I'll turn it over to Shoshana, who will talk about the attitudinal data. So it turns out that when we analyze the grade distributions in specifications grading courses and courses that use traditional grading, so again, typically like 3 exams or so plus a final, what we saw is there is a difference in the grade distribution between those 2 types of classes. And the things that we observe within those differences are that if we look at just the students who pass the course, so again, for these purposes, that's A, B, and C, there was a much higher proportion of students who earned an A. And this could be because students have multiple chances to show that they understand course content. And one of the things that we've observed is that this kind of format works really well for students who really want to be very successful. They want to do really well because they can sort of work on things even if they don't master them the first time. And so it really does encourage and incentivize students to really shoot for the highest grade they can possibly get, because really that's an A for everybody, even kind of late in the semester, assuming you've been keeping up at least somewhat. And so as a result, there were a relatively smaller number of B's and C's within that passing category. If we take a look at the groups of students who were not passing the course, within the specifications grading sections, we had a larger number of withdrawals and D's and a smaller number of F grades. Now, some people might say, well, if you're having more students withdraw, isn't that a bad thing? I think there are certainly positive and negative components to that, but one of the things that I try to keep in mind is that if we are seeing students withdraw from a course, at least it's not impacting their GPA, because that's how our withdrawal system works. If they get a W, it doesn't factor into their GPA. Maybe it just wasn't the right time for them to be successful in that course, given their other courses or given whatever else they have going on in their life. Okay. Yep, so I'll turn it over to Shoshana, who's going to talk about the end of semester assessment questions. Oh no, sorry, that's me. Yes, we had surveys administered at the end of each semester. The course content, she's going to talk about the attitudinal stuff. I apologize. Okay, so sorry, I knew there was one more thing I had to say. So we also compared the student performance on these content-based questions that were on the end of semester assessment. And you can see the distributions between the traditional grading and the specifications grading sections. And what we saw was that the median and maximum scores achieved in the specifications grading sections were higher. And when we did the statistical analysis, what we saw is that there was a small effect size, but a still significant, statistically significant difference in between students who were in traditional courses and specifications grading courses. So this does suggest that hopefully the additional incentivization and opportunity for students to go back and really make sure they understand the course content is being borne out by their actual performance at the end of the semester when they're asked to complete items that show their understanding.
All right, now I'll take over. And I can mess up my own slide progression rather than everybody else's. All right, so as everyone mentioned earlier, there was two hypotheses. One was based on success in the course, but we also wanted to know whether students enjoyed specifications grading, whether they felt that it helped them. So we made some survey questions, again, along with those content questions at the end of the semester. And the attitudinal questions were on a 5-point Likert scale, where if they answered agree or strongly agree, that was indicative of a positive response. And strongly disagree or disagree was indicative of a negative response. And so here we go. It doesn't let me progress. There we go. So this might look like a lot. I'm going to break it down for you. These are the 10 attitudinal survey questions that we had. You don't have to go and read each one of them. I'll point out the things that we want to highlight. We wanted to know whether they were understanding the course content. So there's two questions talking about understanding and retention of course content. So the method in which this lecture was graded either helped my understanding or helped my retention. So if they answered positively, they're saying, yes, I felt like this helped understanding and retention. The next 3 through 9 of the questions were about the course learning objectives. So in addition to the individual learning outcomes, those 20 learning outcomes, we do have a set of course learning objectives that are part of our syllabi that kind of break the content down into cell cycle or gene expression. And so we had an individual question for each one of those. The method in which this course was taught helped me with this concept or that concept. And then last but not least, did the method in which this course was taught promote my enthusiasm for cell biology? And so we had to word these a little strangely because these surveys were given in some sections that had traditional grading schemes and some sections that had specifications grading schemes. So we had to word it the method in which this was taught. But you can kind of see we wanted to know how they felt. Did it help them understand? Did it help them retain information? Did they enjoy it more? Did it make them more happier to learn cell biology in the general scheme of things? And so again, these are those 10 questions. And what I'm showing right here is under traditional grading, the percent of positive answers. So that would be agree or strongly agree. And then I kind of highlighted, because we like to highlight specifications grading, I highlighted in red with the red box, the percent positive responses to those questions for students who are in sections with specifications grading. So again, just highlighting understanding and retention, you can see there's an increase in the percent positive responses. Actually, if you look all the way down the column, the percent positive responses for understanding, retention, all the different learning objectives, they were all increased. And again, at the bottom in 10, the enthusiasm for learning cell biology for the course content was also increased. I do want to note, and we noted this in our publication also, that these results, although they were increased across the board, these positive responses, they were not statistically significant. If you look at the top, you see that there were many fewer responses for traditional grading than specifications grading. And I'll tell you why this is a positive thing. The first semester we implemented this, we had maybe it was 2 or 3 sections where specifications grading, and people had kind of stuck with their traditional grading. But once we did our first semester and got together and talked about how well it went and saw those positive evaluations on our student evaluations and talked to our students, people all switched over. Most of the faculty started using specifications grading. So then once we got that IRB approval to do the surveys, we had very few traditional graded sections left. And we think that's why. And that's why we don't have the statistical significance, but in our hands and from our work with the students, we know that they are definitely feeling that it's helping them and helping them learn and helping them retain information. So kind of in summary of everything we've told you today, we saw increased academic performance on content questions at the end of the semester. In classes with specifications grading, we saw a higher proportion of A's. And definitely positive student attitudes. And even in recent conversations I've had with students, because as an instructor teaching specifications grading, it's a decent amount of work, and it can feel a little stressful from our side also, but they are really truly appreciative of these opportunities and the ability to grow and not just be tested and then forget it, that they have a chance to think about it and see what they did wrong, and then retake those assessments. And so we hope what this will be is a model for cell biology and other courses in the biological sciences. It's definitely not easy to implement in a biology course. I feel like you really have to figure out what content you want to make into each of those learning outcomes and really work on those assessments as a group, which is what we did. So my last slide is tips for implementation, and we can talk about this when we open up this to Q&A and things. But as Janelle mentioned in the beginning, we basically joined forces to develop these learning outcomes. So we took, I think it was either a semester or an entire year of meeting every other week and hammering out not only which parts of the class we felt absolutely had to be included, are there things that we could move aside if we have time for, because specifications grading takes time in the classroom. There's more quiz time and things like that. And it took us a long time to get that wording together and all agree and all be on board. To think about time for retakes, that has been a bit of a struggle. We do know some faculty in other courses that do it all within the classroom, but that can be very difficult. We already have 20 learning outcome quizzes in our lab and lecture components. And we came up with a strategy of having what we call a retake room, where everybody who does specifications grading donates a certain amount of time. So there's 15, 20 hours where students can go to this room and have a proctored exam. And it's taken a lot of work, and we had a lot of different faculty and different disciplines also help with that. And it made it possible, I would say. A strategy for deadlines and limitations, thinking, how many retakes will you allow? Are you going to give never-ending opportunities? Are you going to say at some point, I still want you to learn this information, but it's not going to increase your grade, which is one of the reasons we do have a limit on retakes. One, because cell biology builds on itself. And 2, it's very, very hard to come up with all these different versions of these quizzes. And so it was very difficult to have more than 3 for multiple reasons. And I cannot even see my last thing. Oh, multiple versions. I mentioned that. Sorry, I couldn't see that when I was talking about it. So those are some of the tips and tricks that and things we learned along the way as we were implementing this in our classes. And that's the end of our slide deck. I wanted to say thank you to our hosts for inviting us to present this work and allow everybody to open up for comments and questions.
Thanks very much. That was a really nice presentation. I think you did a great job of covering a lot of details from the manuscript. I have a bunch of
Real
questions.
quickly before you get started, can you unshare the screen and then I'll spotlight all the speakers?
Right. Thanks, Rachel. So I have a bunch of questions, but the audience has asked a bunch of great questions too, and some of them are exactly the same ones I was going to ask. So we'll just kick right into these questions from the audience. So the first one is by Greg, and he actually was wondering early on in your presentation, for a given quiz, are the 3 or 4 questions for each learning outcome, are they short answer? Are they multiple choice? Or what?
I'll go ahead and take this first one and then let somebody else maybe take the next one. We tried to, I mean, we all do a little bit different, but we tried to implement different types of questions. So in general, for example, for one of my quizzes, it might be a 5-question quiz and each might be 2 points, depends on the question. I might have 1 multiple choice, 1 fill in the blank, and then maybe a couple short answers where they can show their knowledge. So I think we all have varied forms of giving these quizzes, but it was definitely a mix of different types of questions.
Let me just do a follow-up. Have you had enough of the questions that you can compare whether the short answer is more effective than the multiple choice, or you still need more data to be able to do that?
We haven't sat down as a group and talked about that. What I will say is I'm still always surprised by how difficult it seems for students with multiple choice. Where I think I'm putting this easy, it's the answers right there in front of you question, they tend to get lost in those a little bit more. And so I've tried, especially this semester, the more and more I do it, to either limit my words, because I've been told I'm a little wordy, so that's not confusing them or getting them caught up, and have them show their knowledge more than in other types of questions.
Thanks very much. So Miriam had a question that was right on top of a question I had about scaling up. So she says that she teaches classes with 100 to 700 students and wonders if you have ideas of how you could do this in a meaningful way with much, much larger classes.
I can try to take that question. It is true that at GGC, we do have small class sizes. I mean, that's just the way our school is built. So we have very small number of students, but we, one of the ways that you can do something like this is do some online sorts of quizzing, especially if your school has, bought some of the proctoring types of software that many schools have invested in since the pandemic has begun. And what's really cool about that is if you get a group of y'all to work together, you can come up with these pools of questions, and then you can randomize which questions they get, or you can make different versions and you can ensure they get different types of questions. So that's one way that you can do that where you can save some class time and they can do some of this stuff outside of class. So that's certainly one way to do it. And I think making pools of questions, that's what we did as a group. We all came up with our own sets of questions, and then we all kind of combine them together. So we have a bunch to choose from, so we can have multiple versions of exams. And I think that's really, or quizzes. And I think that's really, really important. And yeah, so I would say, online quizzing would be a really great way. Or if you do clicker type quizzing, you could definitely do clicker type quizzes in class as the start of the class for your thing. It wouldn't have short answer, of course, but you could do it that way.
Yeah, and I will just build on that when we went in under pandemic mode, it was in the middle of a specifications grading semester. And so we had to very quickly figure out and not change the way we were teaching. And so we made those online initial and retakes, and again, made those pools of questions so that even if they were sitting next to each other, taking these retakes, they might not get the same questions. So absolutely scalable.
A question that was posed a little later on, but directly relates to this is, are your quizzes timed? And how long are they if you do that consistently?
I think we all stick with 15-minute quizzes and, with the combination of maybe 1 or 2 multiple choice, some short answer, a fill in the blank. Almost all students complete the quiz within that time.
Thanks so much. So John had a couple of questions that he posed while you were talking about with the withdrawals, right? So he asked 2 questions right after each other, and I'm going to ask them in reverse order to what he posed. So the first one was, if the DFW rate was similar to what you see in other classes of this size or other 300 level classes.
I can answer that one since one of my hats is aggregating all of the programmatic data, and it is comparable. So our 3,000 level courses, we might have a DFW rate somewhere in the 15-20-ish range, and it was the same in this cell biology class. The only difference being, as Mary Beth pointed out, the distribution within the D's, F's, and W's was different, with more students in the specs grading course withdrawing as opposed to earning a D or F.
Yeah, and one of the kind of hypotheses that we haven't gone in and looked at more is we really think that students have a better understanding of where they are at in the class by withdrawal deadlines. If it wasn't just that they maybe didn't fail, I hate that word, right? The first test in a traditional grading class, and they maybe haven't taken their second yet, or they think it'll be fine. This is very defined. If you don't have this many by midterms, the chance of passing is lower. And so we think that potentially they just have a better understanding where they are at. And I'm hoping that is the kind of thing that accounts for it. And there might be also some students that this might not work for. We haven't really seen that so much in evaluations. We haven't heard this doesn't help me learn and in the survey results, but that could also account for some of the students who it doesn't work for them. Frequent assessments might stress them out too much or something like that.
I think in your conclusions, you indicated that later on you'd like to pursue this and try to sort through it and figure that out. I think that will be a real interesting addition to this study. So David asked, were these randomly selected summative assessment questions drawing from a subset of course learning outcomes?
We went specifically through each of the learning outcomes and made sure there was generally equal distribution across those 20 learning outcomes when we made those assessment questions. It's been a little while since we looked at them, so I don't remember exactly how many for each, but we tried to evenly distribute it across those 20. So
Miriam wondered if this approach decreased the likelihood of cheating among students. And she suggests that maybe at least one indicator of that might be either self-reported or affairs that were incidents that were passed on to university administration for review.
I'm not sure if we kind of kept track of that kind of thing. Anybody else can jump in, of course. You all know that I like to talk, so you can interrupt me. But what I have implemented, and I'm pretty sure a couple other colleagues have, is because we have multiple versions of these quizzes, when we give them out in class and we give them as retakes, students aren't getting the same ones at the same time. So because one of the things I forgot to mention at the end was that we actually had, before we even started specifications grading, come up with 3 versions, right? So it wasn't a last minute run. It was a fun summer of making a whole lot of little assessments. But I think that I'm always giving different versions at different times for different people, and retakes are on different days. And so I think that potentially could decrease, but we don't have the numbers for that or anything.
And I think it's important to mention that, I know, so I use specifications grading in my chemistry courses, and the retakes the students are not allowed to see. Is that the case? Or they're allowed to see them. They're not allowed to take them. They can see them, they can make notes on what they need to work on, but they're not allowed to actually take them. So in that way, the retake versions are secure or more secure. And so that's how we get away with not writing a new quiz for every single student every single time.
So Sean asked the question about how the cell biology material was presented, right? So you started off talking about these different approaches that you use. I wonder if you could give an idea of how they were packaged.
I can jump in and just say that, we're a diverse group of faculty, but I think within our cell biology group, we all use a combination, there'll be some traditional lecture, but there'll also be case studies, problem sets, other types of active learning. So it's kind of a blend of activities, but it's not purely lecture.
And to build on that, yeah, I'll be honest about this one. I feel like specifications grading in the classroom time takes a little bit more of your time because you have quizzes quite often. So I do worksheets and I try to do things, case studies and active learning, but I do feel like I'm losing a little bit of that extra time. And one of the things I really want to work on personally is getting that back. And maybe that involves flipping a little bit, making a few more videos, things like that. So that maybe the basic concepts are videos and then the meat of the stuff is in class and done more with case studies and things. Cause I feel like for me, a little bit, it's dropped off more than I want it to, little less active learning, little more, hey, take this worksheet and we'll talk about the answers when we meet up again. And I always feel horrible about that kind of thing, but.
Yeah, and I'm a flipped, pretty much all my classes are completely flipped at this point. So it took me a while to get there, but I pretty much don't lecture in class anymore. If I do feel like I have to do it, it's because of something that is coming up in the classroom and I go, whoa, stop. And I bring out my slides and I go through it again or whatever. So there are ways that I hold them accountable outside of class for doing those videos. So I do pretty much complete active learning types of things in the classroom. A lot of like think, pair, share, a lot of puzzles, being in a puzzle group, we do things in the body. We've made neurons outside and, been like proteins and stuff like this. So we do all sorts of different styles of learning in my classroom. So.
That's really interesting because earlier John had asked the question about whether some students just couldn't easily adapt to this new learning approach. And he mentioned in the chat that he flipped his classes and he has some students who say they hate the flipped class, right? And it could be different learning styles, or it could just be that students have had so much of the other, it takes a while to adjust, right?
Mm-hmm. Yeah, and oh, sorry, Mary Beth, go ahead.
I was just going to say, I think anecdotally across all of the various subjects in which we've done specifications grading at GGC, it's a pretty polarizing topic. We have a lot of students who will love it and they will seek out courses that use it. And then there are some students for whom they would prefer cram for an exam and then move on. And
the other thing to build on that is that it usually isn't the first time they've seen it when they come to cell biology. We got together as a group because we had seen it be successful in things like chemistry and kind of latched on and used those ideas, but had to figure out how to do it in a biology course. And so it's not usually not the first time they've seen it. Maybe, sometimes depending.
That would be interesting data for us to collect in the future.
Yes. So Idalise asked a couple of questions together. One of them is, when the students took the assessment, did they complete the same assessment or was it a similar assessment of the same learning content? And I think you already said that they're not exactly the same, that they change with each assessment. So
within sections we're talking about, or the survey data?
Retakes. These are
retakes. Oh yeah, it's usually, so we tried very hard to say about 20% of it should cover this, 20% of it should cover this. But the way the questions are asked, or this question might be about microtubules and that one might be about intermediate filaments. And so kind of just, it's the same, not exactly the same content, but within the same learning outcome.
So she also asked the question of, for your first survey question, where you're asking students about their understanding of the material, whether you did a correlation of how well they did in the class with their response to that question.
We did not go back and do individual student lining up student responses with that. And I honestly have to think about whether we can. I mean, I have all the data, probably can go back and look at that kind of thing for sure.
So you dealt with this along the way, but I think one of the issues has to do when you think about numbers and the types of things you're analyzing, you did some pretty complicated statistics. It wasn't just a simple statistical analysis. And I imagine as you break down to more and more detail, the statistics will get even more complicated just because of numbers, right? So, but I'll just point out that you did mention in your conclusions a number of really interesting questions that came out of this that you plan to look into the future. So this isn't the end of the story for the audience. They should be well aware of that. So Maria asked if you had any students who passed all or most of the quizzes the first time through, met all of the earning outcomes quickly, and what did they do with the extra time?
They spent their extra time being happy. So if a student had mastered all of the LOs going into the final exam, then they didn't have to worry about scheduling all of this extra time to do the retake. So they had that time back to either prepare for the final exam or to focus on their other courses. And that was by design. So the students who we were trying to incentivize demonstrating mastery the first time around. So yeah, I think they were relaxed and happy.
They could spend more time studying for Mary Beth's chemistry classes.
Oh yeah. I will say generally our students are co-enrolled in cell bio and organic chemistry at the same time. So they're like, I can go study organic chemistry now.
So I'd like to just shift really quickly. Rachel would like to do a quick survey of the audience. We'll come back. We have a lot of good questions left to deal with, but she wants to do a quick survey. It's really important for us to be able to continue the GMB programming and to make sure that it's tailored right for the audience. So please take a couple minutes and complete the survey.
You for providing feedback, everybody. Stanley, back to you.
All right, thanks so much, Rachel. So we have another question from Sarah, and she wondered if you could give an example of how you broke down your learning outcomes into more specifics. I don't know how easy that would be to do on the fly.
I guess I can try to take this a little bit. So let's say a learning outcome was to like describe, we actually, I don't know, this is another class. So I haven't taught cell in a little bit, but like describe the process of mitosis. Like to us, we may know what that really means. To a student, it could mean a wide range of things because mitosis is kind of like a big idea for them. So maybe what I really mean is I want you to be able to distinguish between each of the stages. So be able to tell me what's going on in prophase, metaphase, telophase, anaphase, whatever. I might also want to know, do you understand what's going on with the chromosomes in each of those stages? Or I might want to know, do you know what's going on with the cell membrane or the nuclear envelope in those different stages? So there may be, I might be looking at wanting to know specific information about the process of mitosis, but just by being kind of general with the student, they may not understand the exact things I'm going to be looking for when I look at their answer. Do I want them to be able to draw it? Maybe I want them to be able to draw it. That's another thing. So to describe it, maybe I want to see you draw it. So going in and saying, describe the process of mitosis. A, I want you to be able to draw each step out, showing me where the chromosomes and what's happening in the cell. B, I want to know what's going on with the cell membrane in each step, or what's happening with the chromosomes. C, you know, like you could, so you would just kind of bullet point each of the individual things that I, as a grader, am going to want to see in an answer that they provide to me. So that's kind of how we went about it, is what exactly are we, when we say this, what are we really going to be grading for and looking for? And we just spell it out for them.
Yeah, and to know, I'm almost 100% sure that we listed our 20 learning outcomes as a supplemental file in our publication. So you can see what we did individually, where it might be something like, originally it was understand the process of transcription in eukaryotes. We broke that down into the players, the enzymes, and the things that bind the DNA, versus actually physically using that ability to transcribe the actual molecule and finding a start site and things like that.
So this comes to a question I wanted to ask, and that is, some of these processes, the basic, if you would, central dogma associated with cellular reproduction, you know, transcription, translation, DNA replication, those are often in cell biology classes and in microbiology classes and in genetics classes and in biochemistry classes. And I know that some of those classes follow your cell biology class in your curriculum. I wonder if you have any idea how they do on that kind of content in subsequent classes.
Yeah, so I can take this. I teach genetics, and it depends. So some students, I think, are able to, they remember going through this stuff in cell bio because they kind of studied it and they're like, oh, I remember doing this. I remember, and they'll even still say it to us in class. And then other students, you know, so cell, I think we do this topic in cell at the beginning of cell bio, and in genetics, I actually do it kind of actually near the end for the way I have mine organized. So many of the students, I think, remember it. Some of them don't. And so, I mean, you know, you can't win everybody. I guess not everybody made an A in that class in cell bio, as you saw from our distribution. But I would say those A students, I think those are the ones that probably, I have now, this is anecdotal, right? I haven't done it, but I think those are the ones that are probably telling me, hey, I remember when we did this. Oh, Dr. Katzman told me blah, 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 blah. Or, you know, so I think it's, there's some that are getting it better and it's sticking with them better. And I think there's some that it's really not, but that's, you know, that's just the way it is.
Well, so we're getting close to the end, but a couple of really important comments, and that is, there are some more questions here. And one of the other issues is that you built this set of learning outcomes based upon some learning outcomes from the American Society for Cell Biology. And, you know, for microbiologists or people in other areas, it might be really useful to have some group think about really good learning outcomes. We hope that you'll come to ASMQ, and people with questions about learning this content and how you can implement it will come to ASMQ. How could you apply this to labs and other types of questions like that? So that is one of the questions, and I'm sure that some of the people in the audience will probably send you emails to ask you more questions, and that's the fun part of being an expert in a field. So one thing I will add to this, and others have mentioned it too, you did a very nice job of showing teamwork and showing how you can answer big questions by working as a team that would be hard to really get the kind of data you need to answer them definitively with small individual groups. And I know that the assessment part of this was really also really critical with the different parts of things. So thank you so much for talking to us today. I know we could have spent another hour on questions, but it was really a fun and educational discussion. So thanks again.
Thanks for having us.
Rachel.
I learned so much. Thank you for sharing, everybody. So the recording of this will be posted to Jimby's YouTube playlist early next week. I just posted a link to the Jimby Live webpage into the chat, and there you can find the registration for the April 8th webinar. It's not April 1st, it's going to be April 8th, and it's going to be called Characterizing Biology Education Research, Perspectives from Practitioners and Scholars in the Field. And that'll be led by Jeremy Su and Stanley Lowe. And these are active individuals in SABRE, the Society for the Advancement of Biology Education Research. And ASM Education and SABRE are going to join forces and offer a newly relaunched Getting Started in Biology Education Research course. And by the time of the April 8th webinar, the registration and the information will be up on the web. So come to the webinar, hear about how you can get involved with learning how to do education research. And also included in that course, you can come to ASMQ for free in the cost of registration for the course. So do consider coming to that April 8th webinar. So
Can I add something, Rachel?
Oh yeah, please.
Also, please tell all of your friends about it too. I mean, expanding the education research community is really done word of mouth very often. So please share that with your friends.
Thank you so much. You're absolutely correct. And it is more fun to have people in the course that you know with you too. And of course, you're going to meet lots of other scholars around the country that do this work. So thank you to our speakers today. Thank you, Stanley, for moderating the discussion. We'll see you April 8th. Bye-bye, everybody.